Okay, thank you very much for the kind introduction. So it's my great pleasure to be here. This is the first time for me in Taiwan, so I'm very excited. I would surely like to thank Dr. Yin, the founder of the Tang Prize Foundation. This is a great honor for me to be one of the awardees for the Tang Prize for Biopharmaceutical Science. I would like again to thank Dr. Yin, because I think it's very important to highlight science to the public, and this prize will surely become more and more important throughout the world. And I would like also to thank the members of the selection committee for having chosen the field CRISPR-Cas9. So dear distinguished guests, dear ladies and gentlemen, I'm going to start with some basics about bacteriology, what is a CRISPR-Cas9 system, some basics about genetics, introduce you briefly to the CRISPR-Cas9 pathway, and give you some insights how this pathway led to what we know now is really a transformative genome engineering technology, CRISPR-Cas9. So I will start with a quote of Jacques Monod, who was a French macrobiologist, and wrote this quote in a book published in 1970 entitled Chance and Necessity, and that is as follows: modern molecular genetics offers no means whatsoever for acting upon the ancestral heritage so as to improve it with new features, to create a genetic Superman. On the contrary, it revealed the vanity of any such hope. The genome's macroscopic proportions today and probably forever rule out manipulation of this sort. The reason why I chose this quote is because Jacques Monod, together with Wolf and François Jacob, worked on really trying to understand gene regulation in bacteria, and had really the concepts that you can find in CRISPR-Cas9, and that are the concepts of bacteria infected by viruses, having regulatory mechanisms that allow to defend themselves against the infection by viruses. They also developed all these notions of operands of genes, of enzymatic reactions, of DNA, RNA proteins, of also RNA being potentially having regulatory function, in addition to be messenger RNA, transfer RNAs, and ribosomal RNAs that are important for the translation of RNA to proteins. It is a little bit peculiar that actually, although Jacques Monod was a visionary, that he wrote this quote, because surely the years following this quote prove that you can manipulate genes and their expression. Here, this is a picture of François Jacob, who worked together with Jacques Monod, and I like this quote of him: nature is a tinkerer, not an inventor. So really this idea that nature is all about bricolage of genes. And this is also what is CRISPR-Cas9 about. So some important aspects of genetics, because we are really in the field of genetics with CRISPR-Cas9. And for those who are not aware of important and selected here milestones towards really the development of gene therapy and genetic tools, the fundamental genetics started in the 19th century with the establishment of certain rules, with Darwin on the origin of species, Mendel on the segregation of alleles, and Michel isolated the DNA in 1871. 80 years later, the DNA was shown to be the carrier of genetic information in 1944. In 1953, the structure of the double helix of DNA was determined, and then in 1966, the genetic code was revealed. And surely the need to understand the functions of genes and unravel really the background and the complexity of genomes in different types of organisms. And this came thanks to a number of enzymes and proteins and systems that were identified in bacteria and also in viruses, and mostly trying to understand how bacteria defend themselves against their own viruses, the bacteriophages. And this started in the 60s, with really in the mid-70s, the democratization of restriction enzymes that are used for cloning and other types of enzymes that have been very useful for the recombination of DNA and in the field of molecular biology and the start of really genetics, the ability to manipulate genes to know their functions and to understand also how the gene expression is regulated. Then you have the sequencing of DNA and the ability to amplify DNA. Other also important milestones, and here I would like to switch to gene editing, whereby studies on a type of nucleases existing in eukaryotes, the meganucleases, homing nucleases, led to the idea of engineering new types of nucleases, zinc finger nucleases and talent nucleases, that started to be used in the beginning 2000s and later for talent starting 2010. Those enzymes are very interesting because they allow to perform precise surgery on genes. They use a mode of action that is protein DNA recognition, whereby a new nuclease has to be engineered for a new type of sequence on the DNA to be modified. So these technologies work well. However, there were some, let's put that way, some bottlenecks with regard to a democratization of this enzyme due to the engineering part of the system. So there was clearly a need for a tool that will be easier to use, less time consuming, and less difficult, so easier to design. And this is really what CRISPR-Cas9 brings. It's a fast and easy tool for the biologists to use in different aspects of genetics. The technology is a two-component system in the sense that you have a protein component and an RNA component. What you see in gray is the DNA, here to be targeted in order to be modified. You have the protein component that is Cas9, that is colored in blue. And this Cas9 protein is programmable with an RNA component that is in nature two RNA components, CRISPR RNA and tracer RNA, linked here together with a linker for the purpose of providing a single guide RNA. And the manipulator just needs to change the green portion of the RNA molecule according to the sequence of the DNA to be modified, and through base pairing of the sequence in green of the RNA component and the cognate sequence on the DNA, the enzyme can be guided to the right place, and the enzyme is using two nuclease domains to cleave one strand of the DNA. And there is a motif that is important on the DNA to be considered, is the so-called PAM, for protospacer adjacent motif. This is a short stretch of nucleotides. In this case, it's NGG for the system originating from Streptococcus pyogenes, and this motif has to be present right downstream of the sequence that is targeted by the RNA molecule. So it's an easy programmable system that works very efficiently, and the biologists around the world use mainly the system originating from Streptococcus pyogenes that happens to be particularly efficient. Now, some words how we came to this system. This is the representation of Streptococcus pyogenes. So these are bacteria that form cocci. They are here colored in red for the purpose of the picture, and they are invading human epithelial cells that are colored in green for the purpose of the picture. Streptococcus pyogenes is also called group A Streptococcus, and it's a strict human pathogen. You find this bacteria only colonizing the host. And you have some of us are colonized with Streptococcus pyogenes without developing diseases. But this bacterium has the ability to cause a wide range of diseases, from mild infections such as pharyngitis or more severe infections such as necrotizing fasciitis or toxic shock syndrome. So in principle, the idea for us to work on this pathogen is that in the lab, we are mainly interested in understanding different mechanisms of regulation that allow to understand how the bacteria can adapt to the human host environment, how they can survive, and how they can cause disease. And to a certain extent, we are interested how the host can react to the bacterial infection, and surely how the host defends itself. So we are focusing, as a matter of fact, on innate immune mechanisms to a certain extent. And you know that we have developed innate and adaptive defense mechanisms against infections with bacteria, viruses, or parasites. And the bacteria can be themselves considered as a host. They can also be attacked as any organism. And in their case, they will be attacked by so-called mobile genetic elements. The mobile genetic elements have different nature. They can be the viruses of the bacteria, so the bacteriophages. They can be plasmids, and they can be transposons. So transposons and plasmids, they can bring to the bacteria new genes to change the features of the bacteria. So they can bring, for example, antibiotic resistance genes. They can also, for certain plasmids, bring genes that allow the bacteria to cause disease more efficiently, so what we call virulence genes. And the phages can enter 2 cycles, roughly. So when a phage infects a bacterial cell, the DNA is internalized into the bacterial cell, and here either the phage enters the lytic cycle, whereby the DNA of the phage will replicate, and there will be production of phage particles that will lyse a bacterial cell and propagate to lyse further bacterial cells, so to really kill bacterial cells. The phages can also enter the so-called lysogenic cycle. They are called temperate phages, and they can insert, the DNA of those phages can then, after invasion, can then insert into the genome of the bacteria as prophages, and in this case, it can bring new traits to the bacteria via acquisition of new genes that can be propagated throughout cell division. So, for example, in the case of Streptococcus pyogenes, you have a temperate phage population that can bring new genes that allow the bacteria to cause severe forms of diseases in humans. Surely enough, the bacteria have evolved systems to defend themselves against mobile genetic elements. You have different systems existing. A lot of them are classified as innate immune systems, and for example, the restriction modification systems that are consisting of recognizing the DNA of the mobile genetic element that will not have been modified by the systems of the bacterial cell and will be recognized and cleave as a foreign DNA element, so that this DNA element after cleavage cannot propagate, is one system that is an innate immune defense system. And this, the understanding really of the restriction modification system, for example, has led to the identification of restriction enzymes that have revolutionized molecular biology, and other types of enzymes come from trying to understand the interaction between bacteria and viruses. You have one system that is the first and only adaptive immune system known nowadays in bacteria and archaea. This is a CRISPR-Cas system, and it's an RNA-mediated system. CRISPR stands for clustered regularly interspaced short palindromic repeats, and Cas for CRISPR-associated. So obviously, there are some repeats in the system. That's why this name contains repeats that are interspaced. So this is the way it works. The CRISPR-Cas system is consisting of 3 steps. A first step of recognition of the phage, because it's an adaptive immune system. So you have a first step of adaptation, whereby the bacteria recognize the first infection with a phage. Then you have the expression of the RNA and protein components, and then you have the interference, whereby the RNA and protein components forming complexes can recognize the phage genome upon a second infection. So the system consists of an operon of genes called the CRISPR-associated genes, coding for the CRISPR-associated proteins. And in the vicinity of this operon of genes, you have a CRISPR array that consists of the leader sequence that contains a promoter region, and that is followed by a series of very short repeats that are interspaced by spacers. So the short repeats are identical to one another, and are interspaced by very short sequences that are here indicated in green. And those sequences are called spacers, and they have as origin mobile genetic element sequences. So in principle, the way it works is as follows. Upon a first encountering with a phage, the DNA of the phage will be internalized into the bacterial cell. Roughly, it's more complex than this, but some CRISPR-associated proteins will recognize the invading DNA, will cleave a portion of the invading DNA, and will insert it into the CRISPR array at the level of the leader sequence, so that it's somehow functioning as a memorization mark. This is the way the bacteria memorize the infection, by inserting a portion of the DNA of the phage into its own genome at the level of the CRISPR array. So those different spacers correspond to different memorized marks of former infection with different mobile genetic elements. Then you have the expression phase, whereby the CRISPR-associated proteins are translated, and the CRISPR array is transcribed into a long precursor CRISPR RNA molecule. Here, the repeats contains palindromic sequences that can form hairpins. And you will have a CRISPR-associated protein that will function as an RNase and will cleave the long precursor CRISPR RNA at the level of the repeats to generate this single guide RNase that each contains a portion of the repeat of the long RNA and a spacer sequence that can each target a certain mobile genetic element. And then you have the interference phase, whereby the single guide RNA associates to a complex of CRISPR-associated proteins and guides this complex of CRISPR-associated proteins to the invading genome of the phage upon a second infection, and you have a complementarity between the spacer sequence of the CRISPR RNA and the corresponding sequence on the phage genome. And one protein of the CRISPR-associated protein complexes that will recognize this base pairing between the RNA and DNA and cleave the DNA, so that the DNA, once cleaved, cannot be repaired by the repair machineries of the bacteria because they are relatively weak. And this is a dead end for the DNA that cannot replicate, and the phage particles cannot propagate. Some words that are important here is that the system has largely evolved, and there is a classification that the latest classification has been published in 2015 that is classifying the CRISPR-Cas system into two classes, class 1 and class 2. Class 1 corresponds to type 1, type 3, and type 4. Type 1 and type 3 are the two systems that were studied early on. And the main difference of those two classes is that so this classification is really established based on the number of CRISPR-associated genes and also the variation of sequences of the CRISPR-associated genes. But you can see here that at the level of the interference, you have a number of genes involved for the class 1 CRISPR-Cas, whereas for the class 2 CRISPR-Cas, you have only one gene involved for the interference step. And the type 2 system that is known now as a CRISPR-Cas9 system was really the first type of CRISPR-Cas studied in the class 2 CRISPR-Cas, and since 1 year now, we do know that there are other systems existing in this class 2, and us and colleagues of ours have worked and are working on those systems to know their mechanisms of action. But here I will focus on the CRISPR-Cas9 system. And before I start, I just want to explain you how we came to this system, and I will go very shortly through the various steps. I just would like to nevertheless talk a little bit the history, because I think it is important. In 1987, a Japanese group found some repeats in the genome of Escherichia coli. However, they did not have any idea of what could be the function of those repeats. This was in 1987, so 30 years ago. Then at the end of the 90s, some scientists found that actually the CRISPR array, by Northern blot analysis, could produce these RNA molecules of different sizes. So there was this idea that there were RNA molecules produced from the CRISPR array, and that most likely processing will be involved because there were different sizes and short sizes of so-called now CRISPR RNAs. Then at the beginning of 2000s and mid-2000s, different groups of bioinformaticians really described that these repeats were interspaced by these spacer sequences of mobile genetic element origin, and that in the vicinity of the repeats you had these genes encoding proteins that had really a lot of homologies with proteins that can interact with DNA, interact with RNA, cleave DNA or cleave RNA. So this was really the first hypothesis that the system could be an RNA-mediated adaptive immune system against mobile genetic elements existing in bacteria, but also in archaea. Just to say that the system does not exist in all bacterial species and archaeal species. You find the CRISPR-Cas system in 50 to 60% of the bacteria and 90% of the archaea. It's just an accessory additional defense system for the prokaryotes. Having said this, this was followed by breakthrough stories that was in 2017 that really showed that indeed the bioinformaticians were right and that this system was really an adaptive immune system in bacteria. This was studied in Streptococcus thermophilus by the group of Horvath, Moineau with Barangu. So really showing that indeed if you challenge Streptococcus thermophilus with phages and that this strain of Streptococcus thermophilus will have the CRISPR-Cas system, you can generate strains that are resistant to phage infection with a concomitant insertion of a portion of the phage in the CRISPR array. So this was the first, how do you say, identification of really that this system is biologically an adaptive immune system. And then a number of colleagues of ours really focused on the type 1 system, the type 3 system. So this started with John van der Oos with Stan Brons on the type 1 system, and also Michael Turns on the type 3 system with Marafini and Santimer, who really started to understand how the system was working. And I hope, I'm sure I forget a lot of people, but just to say that there are some complex histories in the CRISPR-Cas system, that a lot of people have brought their kind of ideas at different steps. The reason why we were interested in CRISPR-Cas9 for my lab, it's because we are always interested in different types of mechanisms that would allow to somehow bring new mechanisms that could be exploited for therapeutic purposes, whether it's to find new anti-infective strategies or new antibiotic strategies. And also, I come from the mobile genetic element field, also this idea that the small RNAs can develop into interference mechanisms. So we were focusing, among other mechanisms, on small regulatory RNAs. And small regulatory RNAs are RNA molecules that can interfere with gene expression in bacteria by either interacting directly with messenger RNA and destabilizing, for example, the messenger RNA, or interacting with the messenger RNA to activate or inhibit the translation, or interacting directly with proteins and activate or sequester them from their functions. What was missing at the time, and when we started to work on these small RNAs, was really a small RNA that will directly target DNA to really change effectively the DNA, and then small RNA that will be important for regulating the expression of another small RNA, and this is really what the CRISPR-Cas9 brings. Now, with regard to tracer RNA, we identified this RNA molecule through a bioinformatics screen, which we performed in 2006-2007, as well as a CRISPR RNA. So we were interested in finding small RNAs in asparagines. We were already working on 2 of the small RNAs, and we wanted to have an idea of more small RNAs. So we found a lot of small RNAs, and some of which were very well expressed. This was a case of tracer RNA. This RNA was interesting because we had found the messenger RNA target that was coding for a virulence factor, and there was a very nice interaction. So we saw that tracer RNA was really involved in the regulation of expression of this messenger RNA coding for a virulence factor. However, we could not make sense of this interaction. But one thing was clear is that this tracer RNA was located in the vicinity of the CRISPR-Cas9 system, also called type 2 CRISPR-Cas system, and that already at the time in 2006 to 2007, this gene Cas9 that had different names prior to Cas9, it was called CSN1, Cas5, CXX12, and that this gene was already hypothesized by bioinformaticians to code for a protein that will have 2 nuclease domains, a RUC nuclease domain and HNH nuclease domain, and this is what is known now as a protein Cas9. Now, this system was clear for us. We had the chance, for sure, to work with asparaginase, and we had the chance that we had this system in asparaginase. this system is really the minimal system, and when we started to work on the system, there was no really insight into the molecular mechanism of the system, and we tried to tackle. So we were interested in CRISPR because we found CRISPR RNAs, and for us it was interesting because this CRISPR system in asparaginase targets temperate phages. So in principle for us it was somehow the identification of a system that potentially could have a role in limiting the acquisition of phage sequences, and by this mean limiting the acquisition of genes that code for factors that allow bacteria to develop more severe diseases in humans. So it was a way to link CRISPR to virulence in the bacteria. So we were working on those two pathways in parallel until the point came that tracer RNA, we were not going anywhere with our hypothesis of targeting messenger RNA. And the idea was really to look whether tracer RNA would not have an interaction with the CRISPR-Cas9 system. The idea came from my side because if you work on small RNAs in bacteria, you know that they can be located in cis and work in trans. They can be located in cis of their target and work in cis. They can be located in trans of their target and work in trans. But you know, actually nothing was known about the regulation in general of the expression of CRISPR systems. So the idea was maybe tracer RNA has a role in this matter. So we really used a genetic approach and the biochemical approach to show this pathway, which I'm going to describe very fast. I just want to say here that we really developed tools that not a lot of bacteriologists even in our days are using, that are really tools of precise gene surgery in bacteria that really allow to perform knockouts in frame deletion fashion with no polar effects, which was very useful to really make sure that we were certain of the functions of the different genes we were knocking out in this different pathway, and that now CRISPR-Cas9 it's maybe easier to use to knock out genes in human cells, and we are still left with our tools to perform precise gene surgery in bacteria that are a bit more labor-intense to use, but at least we are happy that CRISPR-Cas9 the understanding of this pathway in bacteria has facilitated now the genetics in higher organisms. So the way it works is as follows. Tracer RNA contains an anti-repeat sequence that allows it to base pair with each of the repeats of the precursor CRISPR RNA molecule. So it forms a duplex at the level of each repeat. And this it's recognized by the protein Cas9, at the time it was called CSN1, and it allows to stabilize this duplex RNA. And then it's recognized by an enzyme from the host, from the bacteria, that will cleave the precursor CRISPR RNA molecule and also the tracer RNA molecule at the level of the interaction. So this was new compared to the complex systems I showed you of the class 1 that rely in general on only the CRISPR RNA molecule. And at this step, one protein encoded by the Cas system that can take the function of really processing the precursor CRISPR RNA. Here it's different. You have Cas9 involved, but just to stabilize the duplex RNA. Then you have tracer RNA and an enzyme RNA3 from the bacteria. Then what happens is that if you cleave, you produce an intermediate form of CRISPR RNA with a repeat in 5', the spacer in the middle, that can target the phage, and then the repeat sequence in 3' that can still base pair to tracer RNA. Next mechanism whereby we still don't know the nature of the ribonuclease, but in principle what is clear from also RNA-seq analysis, which we had at the time, is that the final product of the mature form of CRISPR RNA is spacer in 5' and repeat in 3'. So one needs to mature further the RNA after the first cleavage within the repeats, and this is most likely taken care of by a ribonuclease from the bacteria, whether it's an exo or endoribonuclease or two actions of an exo and endoribonuclease. So the final product is as follows. You have the mature form of CRISPR RNA and you have the tracer RNA form. Surely, what we described at the time is that tracer RNA was important to trigger the maturation. What needed to be shown, and maybe it was not really so clear actually, but the idea at least was that tracer RNA may be involved in really the last step of interference through this base pairing and stabilization of Cas9. And this is what we showed using also a genetic and biochemical approach. So what happens is that this complex can scan the DNA to first find the NGG. So Cas9 is a protein that has a domain that allows it to first bind to this protospacer adjacent motif. Then you have R-loop formation, so an homology search, so that the portion of the CRISPR RNA that can form base pairing with the DNA so this RNA molecule is going to look for the cognate sequence on the DNA. You have R-loop formation, and Jennifer will explain you how it works. I hope, I'm sure she will go more in the details of the structure, but you always have a change of conformation of the protein each time that it binds the RNA. It binds the DNA, and then you have the homology search with the CRISPR RNA. And ultimately, you have the two nuclease domains that are well positioned to cleave the DNA in a blunt fashion, and you have double-stranded DNA breaks that are then produced. So here you have this a little bit more in details. Okay, I don't have a picture of the assay, but what we showed, at least the first experiment that could really show that it's a complex of two RNAs with one protein cleaving the DNA, was really to a simple cleavage assay, like you will do a restriction enzymatic reaction when you have the protein, the two RNA in the test tube, and the DNA, and you can see that indeed the final reaction is a blunt cleavage with two cleavages here possible that occur 3 base pairs upstream of the PAM. That is in this case, is in specific case, 2 possibilities of NGG, so AGG and GGG, that's why you have two cleavages. Here, something that is important to mention is that the PAM sequence is really an element that allows to recognize self and non-self, so that the system can recognize the phage, because in the vicinity of the sequence that is targeted by the CRISPR RNA, you have the PAM sequence that is not present on the CRISPR array, so that the system cannot recognize itself. And this PAM sequences was identified informatically by colleagues of mine, and really biologically with the work of Sylvain Monod, which specifically for the system could show that indeed the PAM sequence was important to really allow the CRISPR-Cas9 system to work in vivo and have a DNA blunt cleavage. And this was a study that he published in Nature in 2010, before the tracer RNA was identified and before all the molecular biology and really genetic experiments to really focus on the mechanisms were published. So what happens when you have a double-stranded DNA cleavage, you have exactly what zinc finger nucleases and talon nucleases can do in a sequence-specific manner. They can also introduce double-stranded DNA cleavages. And this will be taken care by the different repair mechanisms in the higher organism, in cells and organisms of the higher higher organisms, and this will actually introduce this will trigger different pathways. So either non-homologous engineering repair mechanisms whereby you can introduce smaller or larger deletions at the site of the break, or you can introduce an homology donor into the cell, and designing the homology donor in such that you have upstream and downstream of the sequence that is used as an homology donor, homology arms, so that by a double crossover you can replace a sequence of interest by another sequence of interest, which allows you to also introduce mutations and perform different modifications directly on the DNA. Something here that is really, how do you say, a proposal which we published in the journal Science in 2012 together with Jennifer, is that the system was obviously a simple system to use. Obviously, it was really attractive as an RNA programmable system. and that with the idea that it could really be a system that will be an alternative system to zinc finger nucleases and talent nucleases to perform genome editing in cells of the three kingdoms of life for biotechnological, biomedical, and gene therapeutic purposes. The system has several advantages, and everyone agrees to say that it's cheap and easy, it's efficient, it's versatile. I'll show you some examples. It allows multiplexing in the sense that you can design different RNA molecules to target different sequences at once on the genome. The system is highly specific, even though for sure in nature you never have 100% specificity, but a lot of our colleagues, specifically in the Boston area, but also throughout the world, also really develop different tricks to really improve the specificity of the system. The system has low toxicity, and among the challenges to really bring this technology to direct applications in medicine, really the need to develop in parallel to improving the specificity of the system, really the delivery of the CRISPR-Cas9 into the right cell and the right organism. About a little bit more than 6 months after we published our paper in Science, a number of articles were published around the same time, showing that the technology was really useful to perform genome engineering in human cells, and this we started with a group of Tang, and I'm sure he will talk to you about the different aspects he has studied since then, the group of George Church, and then others followed. Then also the possibility to perform knockouts in mice and multiple knockouts in mice, the possibility to use a system in bacteria, in plants. Very interestingly, very fast as well, what came is that the technology was very useful to really improve the genetic technologies for all the scientists working on model organisms such as Drosophila, yeast, zebrafish, and then also showing that it's useful for human organoids and that it can also work in monkeys. So here you have different aspects of the toolbox, and you will hear this later, I'm sure. So in principle, you can, depending on how you design the system, you can create small or larger deletions and rearrangements on the genome. You can insert new sequences and replace sequences by other sequences. You can use the property that Cas9 has 2 nuclease domains to really mutate this Cas9 protein, and that will not have the possibility to cleave the DNA, and you have an RNA programmable protein that can bind the DNA. So this ability to somehow mutate the domains of Cas9 were surely came from bioinformatic analysis showing that there were 2 nuclease domains, and were done by us, Jennifer and me, and also Sixnis, who also showed that you can mutate these nuclease domains. But in principle, you can have a dead Cas9 that can then be fused to different effector domains, and that can be guided to the RNA to the site of infection on the DNA, and you can use different effector domains that would allow to activate transcription, inhibit transcription. That's why you have a lot of screens that have been developed for using CRISPR-Cas9 for manipulation for modulation of gene expression. You have also the possibility to study epigenetic marks by using as effector domains that can modify the DNA, or you can also label the DNA by using as effector domains these fluorescent proteins. An aspect that is critical here is that, and I'll just show you 2 slides. We surely started with Streptococcus pyogenes in my lab, but when we really identified tracer RNA, the first thing we did is to look at other bacterial species that had this type 2 system, so highlighted with CSN1 at the time. Now we know that it's Cas9 gene. And we showed that actually tracer RNA was present, always associated with the type 2 system. And we found this system in more than 350 bacterial species. So the system is somehow working in a similar fashion, using the same mechanism. However, you have a large diversity of sequences at the level of the RNA molecules, so the repeat sequence, for sure the spacer sequences because they correspond to mobile genetic elements, but also tracer RNA sequences. You have a large variety on the sequence that is coding for the Cas9 protein. So very little percentage of amino acid identity when you compare the different sequences of the Cas9 proteins. Some conserved domains, like an arginine motif that is important for the R-loop formation with the CRISPR RNA. And you have also the nuclease domains that are conserved, but some, for example, the domain that is important for recognizing the PAM on the DNA is different depending on the Cas9 proteins, because the PAM sequences can also differ among the different systems. So you have a large variety, and we started a bioinformatic analysis that allowed the classification of the CRISPR-Cas9 system looking at different and producing in a way different phylogenetic groups based on the Cas9 sequences, where we also identified a long Cas9, a shorter Cas9. There is an interest with a shorter Cas9 because if you do genetics specifically in bacteria, you always want to work with small systems, but also in higher organisms. Interestingly enough, all those systems use different PAM sequences. They also have diversity in the structures of the dual RNAs, but conservation of the structure of the dual RNA within one phylogenetic group. And they use different rules of interaction. What is interesting to say here that there are very few natural Cas9 proteins that work very well, and asparaginase is really the one that seems to work the best. Other proteins are not always working well if you try to implement them in genome engineering in higher cells. So this was our luck to work on strep pyogenes. Having said this, you have a lot of scientists surely working on the engineering of these proteins to have them shorter and more specific. You all know that the field of applications of CRISPR-Cas9 is really broad, ranging from human medicine to synthetic biology and agriculture. In human medicine, I can just say a few words. It's very useful to really, let's put it that way, boost the understanding of functions of genes, because now you have the possibility to somehow modify the genes of human cells and other organisms that are used as models of diseases more efficiently. Really the possibility to also develop all these screenings for new targets for therapeutics, these humanized animal models that are important for the screening for new targets for therapeutics, but also to validate therapeutics that are under development. Also very interesting with regard to bed-to-bench approach, where you have a lot of effort in sequencing of human genomes of patients to find potential mutations that will be responsible for certain diseases. So the ability to go back to the bench and test whether really those mutations are indeed responsible for those diseases. And then the challenge that is really to develop CRISPR-Cas9 as a direct gene medicine in human gene therapy, whereby really CRISPR-Cas9 is brought in an ex vivo approach combining gene and cell therapy, or an in vivo approach with delivery systems directly into the patient, and really having the possibility to treat severe genetic disorders. Among the different type 2 systems, you have others, and my colleagues, I'm sure, will talk about, maybe briefly mention those minimal systems. I just want to talk about CPF1, showing that Cas9 was already minimal in the sense that you have only one protein. However, this protein is guided by 2 RNAs and requires RNase 3 from the bacteria for the maturation step of the CRISPR RNA. The type 5A system is even more simple, and this is interesting in terms of evolution. In this system, you have only one protein. You don't need another RNase, and you don't need a tracer RNA. The protein is doing everything, and very briefly, here this is the CRISPR RNA that forms palindromic, that has palindromic sequences at the level of the repeats, so forming hairpin structures. CPF1 has 2 nuclease domains that allows to recognize the hairpin structure of the repeat of the long precursor CRISPR RNA. CPF1 will use the RNase domain to cleave the repeats of the RNA. And then you have a production of intermediate form of CRISPR RNA that will be further processed, most likely by RNases from the host. But in principle, the CPF1 still bound to the mature CRISPR RNA at the level of the repeat will then be guided to the DNA And we'll cleave the DNA using different modes of cleavage that is not a blunt cleavage, but a cleavage that produces 5 nucleotides far from overhangs. So it's just another diversity of the system, and you have multiple diversities of recognition, cleavage first of the RNA for maturation of the RNA, and recognition of the DNA targets. But CPF1 as a system targeting the DNA is maybe the most simplified in nature. A lot of my colleagues also work on these adaptation mechanisms, whereby you have interesting enzymes also important for cleaving DNA and inserting pieces of DNA into the array, so recombining sequences. So this can be also useful maybe to find interesting enzymes with new biological properties. And I would like to finish to just mention the different institutions where my team and myself have performed the work. So the University of Vienna with Max Efpert's laboratories, then really the project developed in Sweden. So the idea really to combine tracer RNA and CRISPR RNA, and I would like to thank the Laboratory for Molecular Infection Medicine Sweden at Umeå University, that is a partnership with EMBL in Heidelberg. And in Germany, I moved 3 years ago to the Helmholtz Center for Infection Research in Braunschweig, with also an affiliation with the Medical School of Hannover. And I left recently to join the Humboldt University in Berlin and the Max Planck Gesellschaft. And this is the team. So part of the team during our time in Braunschweig, and I would like to thank different people who participated in the CRISPR-9 work, specifically Christophe Schilinski, Elitsa Delcheva, and Ines Fonfara, Anaïs Lerin. And for CPF1, Ines Fonfara, Hagen Richter, Maida Bratovic, and Anaïs Lerin. My collaborators for the part of understanding the maturation of the CRISPR RNA of the type 2 system, the group of Jörg Gogel with Cynthia Sharma in Würzburg in Germany. And then for the DNA interference, the group of Jennifer Darna, and you will hear about the work that she's going to present with Martin Jinek, who has now a group at the University of Zurich and is focusing on the structural aspect of the protein. And then you have the part of evolution that was studied in terms of bioinformatic analysis at a later stage with a group of Eugenie Kounin and Kira Makarova, and 2 biotech companies that I need to cite for conflict of interest. And I would like to thank you for your attention.
Thank you, Emmanuel. Next, I would like to introduce our second speaker, Professor Jennifer Doudna. Jennifer was born in Hawaii, but attended Pomona College in Southern California and graduated from Harvard Medical School in 1989. She has started her career as an independent PI from Yale University, and she was an investigator of Howard Hughes Medical Institute since 1997. Since 2002, she moved to UC Berkeley and UCSF. She's now currently Executive Director of Innovative Genomics Initiatives at UC Berkeley and UC San Francisco. She's also the head of Division of Biochemistry, Biophysics, and Structural Biology in UC Berkeley, and Li Kai-shing Chancellor Chair in Biomedical and Health Sciences, UC Berkeley. Just like anyone else in this panel, she also won a number of awards. I'm not going to read everything, but just a few. As I said earlier, she and the other 2 awardees was also granted Canada's Gardner International Awards, Masry Prize, Gruber Prize in Genetics, and so on and so forth. Jennifer, let's welcome Jennifer Doudna.
Well, good afternoon to everyone, to our distinguished guests, and to Dr. Yin and the members of the selection committee. It's a truly great honor to be here and have an opportunity to share a little bit of our science with all of you. It's been an incredible visit so far to Taiwan. It's my second time here visiting your country, and it's been a very enjoyable opportunity to learn about a little bit about culture and about your science, and to have an opportunity to interact with incredible high school students as I did 2 days ago. Very, very impressive. So what I want to do today is to basically pick up where Emmanuel left off. I'll say a little bit about our work together, the work that we did to understand the function of the Cas9 protein, which, as you heard in her talk, is part of the CRISPR system, an adaptive immune system in bacteria. And then I want to tell you about what we've been doing to understand how this incredible little molecular machine actually works, and where I think it's going as a technology and what it means for us in the future. So I thought I would just start by pointing out that ever since the discovery of the structure of DNA in the 1950s, scientists have been really asking themselves, would there ever be a way to rewrite the DNA code? So of course DNA in cells is a double helix, and it's made up of a 4 chemical letter code. And we understand from a lot of work over the last several decades that many genetic diseases, and of course everything that we know about biology, stems from this amazing molecule. And so what if it were possible to actually change the code, not in a random way, but in a very precise fashion? And I think one of the things that's very interesting about the CRISPR-Cas9 story is that this technology using this immune system in bacteria for genome engineering came about through a curiosity-driven scientific project that was aimed at understanding something very different than where it ended up. So we, as you heard from Emmanuel, we were interested in understanding how bacterial cells fight off viral infection, and in the course of that research realized that we had come across an amazing protein that could be harnessed for a very different purpose. So just to pick up where Emmanuel left off, so as she explained to you, we understand from the work of many laboratories over the last decade or so that the CRISPR systems in bacteria operate as adaptive immune systems. And what they do is to provide cells the ability to detect foreign DNA that is injected into the cell during a viral infection or comes in through a plasmid transformation, to incorporate little pieces of these foreign DNA molecules into the CRISPR locus in the genome, and then generate an RNA copy of those stored viral sequences that are flanked by repeats in the CRISPR element, to use these as molecular guides that can direct Cas proteins encoded by the CRISPR-associated genes, in the form of protein RNA complexes that interact with foreign DNA by base pairing, so RNA-DNA hybridization. And so again, just to build on what Emmanuel told you, so this is representing a bacterial cell, and in the genome of cells that have a CRISPR system, they have an array of sequences shown here as a series of repeats that flank unique sequences coming from viruses. And upon transcription, those sequences are turned into an RNA copy that starts off very typically as a long transcript that has to be processed into individual RNAs that each include a sequence that is derived from a virus. And then those RNAs assemble with one or more CRISPR-associated Cas proteins to form RNA protein targeting complexes that can interact with foreign genetic elements, very typically DNA, but as I'll tell you today, not always. And when that interaction occurs, then these proteins can lead to degradation of the foreign nucleic acid. And so my lab has been involved in, we've been really interested in really all sort of aspects of this pathway, but today I'm going to really focus on our work to understand this part of it, which is namely, how does RNA-guided DNA cleavage actually work? And as Emmanuel explained to you, and this is taken from a very recent publication, a recent review in Science Magazine, we understand that these systems have many different variations in nature. One of the things that makes them so fascinating. And this is showing, again, very similar to what Emmanuel told you, that if you look at the different sets of CRISPR-associated proteins found in these different CRISPR systems, they've been classified into broadly two types of systems. And up here in the class 1s are CRISPR systems that involve multiple proteins that assemble into an RNA-guided complex that is able to recognize foreign DNA and lead to its cleavage. In the class 2 systems, these are all systems that have essentially a single protein that is capable of RNA-guided targeting of foreign nucleic acids. And we've been interested in, sort of studying this fascinating variation in nature, and have over the years in the lab, my lab, looked at the structures and functions of a number of these different types of complexes. But it was really through an interaction when I met Emmanuel in 2011 that we teamed up to understand the molecular function of this protein right here, Cas9. And as she explained, the interesting thing about this was that at the time it looked like a fascinating way that nature had figured out how to stuff a lot of activities into a single protein that might be capable of RNA-guided DNA targeting. And at the time, nobody understood how that worked. And so through our collaboration and the work of Martin Jinek in my laboratory and Krzysztof Chylinski in Emmanuel's lab, they figured out using biochemical methods that we could purify the Cas9 protein, shown here in blue in this cartoon, and show that it's actually a dual RNA-guided DNA endonuclease. And that means that it's basically a protein that has the ability to bind to two separate RNAs, the CRISPR RNA here that contains the guiding sequence that can base pair with DNA. And as we figured out in the course of our collaboration, the tracer RNA that Emmanuel had previously shown to be important for CRISPR RNA processing turns out to also be critical for this DNA targeting activity. So it's a two RNA system that interacts with the protein to form this surveillance complexes. And the way the protein works is it has two chemical cleavers, two active sites that can cut the two strands of the DNA helix by first unwinding the DNA in the region of the targeting. And importantly, this targeting interaction requires that the target site be adjacent to a small motif in the DNA called the PAM. And I'll say a little more about that shortly. And when these interactions occur, then of course the DNA is opened up and it can be cleaved in the manner that Emmanuel described. And so by understanding how this worked, we then realized that we could actually simplify the system compared to what nature had done by creating a single guide form of the RNA that would include the targeting sequence here on one end and the Cas9 binding information over here. And that created a two-component system in which a single protein could be guided to DNA sequences to generate a double-stranded break in the DNA, and it could be reprogrammed by simply changing this short sequence in the RNA right here, something that any sort of molecular biologist it would be a very comfortable thing to do in the laboratory. So we were very excited about this because we realized that it was truly a programmable protein that could be directed to different DNA sequences to introduce a double-stranded break by simply changing this short sequence right here. And I just wanted to show you one of my favorite experiments, maybe one of the simplest experiments we've ever done in the lab, but it was really exciting when you saw the results. And this was work that Martin Jinek did using purified Cas9 protein and taking a plasmid DNA molecule to which he designed 5 different versions of a single guide RNA that would allow recognition of these sequences in the plasmid marked by the red bars. And by incubating the Cas9 protein with one of those different guide RNAs and the plasmid DNA, we allowed digestion to occur and then added a second restriction enzyme that cleaved the DNA a short distance away. And then we could analyze the products of the doubly digested DNA on an agarose gel. And I don't know if you can see this here, but basically each band in cleavage product of the plasmid resulting from these reactions is a different size corresponding to the size that Cas9 was directed to cut by these single guide RNAs. So it was really that experiment that showed us that we had a two-component system that allowed site-directed double-stranded cleavage of DNA. And so the way it works is that it turns out that this is an amazing protein that has the ability to bind to RNA and unwind a DNA double helix. And what I'm showing you here is a 3D printed model that is actually based on a crystallographic structure solved in Martin Ginnick's lab that shows the protein in white holding on to the guide RNA in orange and interacting with a DNA double helix. And when this protein RNA complex finds a sequence in the DNA where there's a 20-letter match to the RNA guide, the DNA is unwound inside the protein to create an RNA-DNA hybrid that enables the cleavage sites in the protein to swing into place and cut the DNA. And as Emmanuel explained, the reason that this turns out to be very useful as a technology is that many labs had shown over the years that when plant or animal cells experience a double-stranded break in their genome, something that happens all the time due to radiation and during cell division, cells have elaborate pathways for repairing these double-stranded breaks, often by introducing a small change into the DNA at the site of the break during repair, or by incorporating a new piece of DNA into the DNA at the site of the break. And so many labs had appreciated that this strategy of introducing a site-directed double-stranded break into DNA could be a great way to conduct genome engineering. And this was a tool that really built on previous platforms, but became widely adopted because it is so fairly simple and easy to employ. And I want to show you one very short video here that illustrates how we imagine that this bacterial system operates when it gets inside the nucleus of a eukaryotic cell. And so we're zooming into the cell. So of course, in a plant or animal cell, the DNA is inside the nucleus, and furthermore, it's packaged in the form of chromatin. And so you can see the blue DNA actually wound around green histone proteins. And somehow this bacterial enzyme has to search through all of the DNA inside the nucleus of a cell to find in principle, a single sequence that matches the sequence of the guide RNA. And when that occurs, then the DNA unwinds inside the protein, RNA-DNA hybridization occurs, the double-stranded DNA break happens, and then those broken ends of the DNA are handed off to repair enzymes in the cell that can seal the breaks, sometimes by incorporating a new piece of DNA at the site of the break. And so it's really this use of this system that's proved to be a very powerful way to create precise and accurate changes to the DNA inside of cells in virtually any organism where it's been tested. So why is, people ask, why did this technology take off as quickly as it did? And I think it really comes down to three things. First of all, the power of base pairing. So RNA-DNA hybridization is a mechanism that cells have been using in many different contexts for genetic regulation. And this system takes advantage of the ease of changing DNA recognition by simply changing the sequence of a guiding RNA, rather than having to change an entire protein for DNA recognition, as was required for earlier genome editing technologies. Of course, you know that there are many applications in animals and plants. And I think what's particularly interesting here as a technology is that this has proven to be a very plastic sort of system. In other words, it's been quite amenable to different kinds of modification, as you saw in one of Emmanuel's summary slides. And that means that in addition to cleaving DNA and introducing permanent changes to a genome, it's also possible to use these systems for controlling transcription, changing the levels of proteins that are generated from a particular DNA sequence, and also for imaging particular sites in a genome inside of a cell. So I want to now move on to telling you a bit about what we understand about the way this actually works. I've always been fascinated by molecular mechanisms and figuring out how molecules function. And so when we started thinking about Cas9, one of the key questions that arose was, how does this protein interact with double-stranded DNA? What really drives DNA target recognition? And one of the things that's worth thinking about is the fact that this is a protein that has the ability to unwind 20 base pairs or more of DNA without any external energy source. So it's not hydrolyzing ATP, for example, to drive that unwinding. So what drives it? I think it's an interesting question, and I'll just share with you what we currently understand about how this works. So one of the experiments we did early on to start looking at how Cas9 searches through a big piece of DNA to find a matching target sequence was in a collaboration with Eric Green's lab at Columbia University. And we took advantage of a single molecule system that Eric had developed for looking at individual phage lambda DNA molecules, these individual green strings that you can see in this slide here, and looking at how labeled versions of the Cas9 protein interact with the DNA in an RNA-guided fashion. And those experiments showed us that really three things. First of all, DNA binding begins at PAM motifs, not with actual base pairing, but really by interacting first with these little PAM motifs, which for this protein is a GG dinucleotide. Secondly, we found out that upon binding to the PAM, the protein is somehow triggered to unwind the DNA, something that we're still really trying to understand the relationship between. What does PAM binding do to the protein to really trigger DNA unwinding? And finally, we found out that there's very high binding affinity between the Cas9 RNA complex and the DNA product, meaning that even after cutting the DNA, it holds on very tightly to those cleaved ends, something that I think is very interesting in the context of thinking about how this works and how it hands off those broken ends to repair machinery in the cell. So in work that we did with the lab of Eva Nogales at Berkeley, we used a combination of crystallography and electron microscopy to begin looking at the structures of Cas9 proteins in different states of assembly with nucleic acid. And this is a cartoon that is based on some electron microscopy work done by David Taylor and Sam Sternberg, two students in the lab at the time, who showed that we could see even at very modest resolution that the protein undergoes a large conformational rearrangement when it goes from the protein alone over there on the left. I think my pointer is out, by the way, but I'll try to direct you to where I'm looking on the slide. So the complex on the left represents the protein alone, and then upon binding to guide RNA, we could see a channel opening up in the center of the protein, which is where the RNA-DNA hybrid winds up once this associates with a DNA substrate. So I'm going to show you now a movie that is based on a series of crystallographic structures solved in our lab and others to look at the state of the Cas9 protein in these different conformational forms. And what you'll see is a really remarkable structural change that happens as the protein assembles with nucleic acid. And I want to say that this movie was made by an undergraduate student at UC Berkeley, Aubrey Thompson. And so when we start the movie, you'll see we're starting off with Cas9 in the unliganded state, in other words, the protein alone. And this is morphing it to the structure bound to the guide RNA, a structure solved by Fugo Zhang and published in my lab, from my lab last year. And you saw that very big rearrangement in the structure that opens up a channel in the center of the protein where the guide RNA is located. And then an additional structural change happens when this complex assembles with a double-stranded DNA substrate. And this is based on both crystallographic and electron microscopy structural data from Fugo Zhang and David Taylor in a collaboration with Eva Nogales, published earlier this year. And so here are the nucleic acids, and you can see in orange is the RNA, in magenta is the DNA, and you can really get a sense of the open structure of the DNA and then these two cleavage sites that are positioned close to the two active sites in the protein for cutting. And so this just gives you a sense that it's really, I think, interesting to think about this as a little molecular machine. It really truly is a protein that undergoes amazing structural rearrangements that allow it to hold on to guide RNA, grab onto DNA, unwind the DNA, and then position the sites in the DNA for cleavage. And one thing that we now appreciate is that this protein really requires that the ability to interact with the nucleic acid to trigger these conformational changes and to enable it to cleave the DNA only when it is truly engaged on a DNA molecule that matches the sequence of the guide RNA, something that helps ensure accuracy in DNA targeting. I wanted to briefly show you this structure here. So this is a sort of a static image of what we call the R loop, which is the structure formed when Cas9 protein and its, in this picture, green guide RNA bind to double-stranded DNA. And what we found was then and only then, when it's really engaged on a double-stranded DNA substrate, does this domain right here, one of the active sites of the protein called HNH, actually swing into place to cleave the DNA, something that hadn't been captured in previous crystallographic structures because it really requires the presence of both strands of the double-stranded DNA to trigger that final essential conformational change that activates the enzyme. So in thinking about how that structure so we understand something about the structural changes of the molecule, how does this actually interact when it's inside the cell? And again, going back to work that we work with Sam Sternberg and Cy Redding in Eric Green's lab, showing that using this single molecule approach, we found that the protein has an amazing ability to bind and release DNA. So it looks like rather than grabbing onto DNA at one site and sliding along to find a target site, instead it seems to work by binding and releasing DNA very, very quickly and slowing down only when it encounters a PAM sequence, which of course occurs fairly frequently in the genome. But that interaction with the PAM somehow triggers a slowdown in the release of the complex to give time for interrogation of the adjacent DNA sequence for a match to the guide RNA. And if there is RNA-DNA base pairing that occurs right next door to the PAM, DNA unwinding begins, and it proceeds as long as there is complementarity between this DNA strand and the guide RNA, eventually forming a full R-loop type structure if there's a full complementary match, and positioning the active sites in the enzyme to generate a double-stranded DNA break. And we've also been curious about how this operates inside living cells, and in a great collaboration with the lab of Robert Tijan at Berkeley and several people at the Janelia Farm Research Campus, we used high-resolution imaging to visualize individual particles of Cas9 that were fluorescently labeled inside living cells. So this is we're looking here at Cas9 particles moving around inside live mouse cell nuclei, and their ability to interrogate DNA as a function of being programmed with different types of guide RNAs. And what we found was that we could really observe a very pronounced change in the kinetic behavior of these particles based on the type of guide RNA that they contain. So for Cas9 proteins containing a nonsense guide that didn't bind to any sequences in the genome, or using a guide RNA that has mismatches right next to the PAM sequence in a target site, we saw very fast kinetics of these particles moving around the cell versus slowing sort of a population of these particles that have much slower kinetic behavior when they have a guide RNA that has an almost perfect match to a target site or a perfect match to a target site. And these are, in this case, we don't have resolution of individual binding interactions with the DNA, so we used a guide RNA that allows recognition of a highly repetitive element in the genome so that we can see changes in the population of these particles as they are programmed with these different guides. So I just want to say briefly what I think are some interesting challenges ahead for really using what's clearly a powerful technology for applications in human therapeutics. And to me, I think the three really important bottlenecks ahead are delivery, as Emmanuel mentioned, controlling the way the DNA is repaired after cleavage for those applications that involve actual covalent changes made to a genome, and also the ethics around delivery and safety and how we think about using this, especially if we think about using it in the human germline. So I'll very briefly mention some of the work that we're doing in the lab currently. So we've had a great collaboration with Roche Biopharma and the scientists there, and this has really been spearheaded by Brett Stahl, a postdoc in my lab, to think about how we might actually deliver Cas9 into cells in a selective way. And in one example that we're currently working on, we've been thinking about how to deliver Cas9 into the brain, a very challenging problem. And the strategy that we're taking as biochemists, we thought, could we just take advantage of what we understand about these RNA protein complexes to engineer them for delivery? And so what Brett and his colleagues in the lab have been doing is to assemble Cas9 RNA protein complexes. So we take purified Cas9 protein, bind to it guide RNAs that are targeting sites of interest in neurons in this case, form RNA protein complexes in vitro in the lab, and then use these to introduce them into either cells that are growing in the laboratory, in this case neural progenitor cells, NPCs, or by directly injecting them into various regions of the mouse brain. And this is, we're using a nice system here. It's actually a reporter mouse that has a gene engineered so that we can detect editing using a positive screen. And the way this works is that we have a reporter that turns red when it's expressed. And initially in these mice, this expression of this reporter is turned off because there are several stop codons engineered upstream of this gene. But by designing guide RNAs that allow cleavage at 3 sites upstream of this reporter gene, this tomato gene here, we can observe that if editing occurs, we remove these stop codons and we can see expression of the reporter. And so what you can see down here is this sort of just showing you an example of when when this reporter is turned on by editing, we can actually see cells lighting up as red, or this is in the brain, and this is actually in cells growing in culture. And the exciting thing is that by using this to then introduce into the mouse brain in different places in the brain, and then staining cells, these are tissue samples stained with different antibodies that recognize proteins expressed only in particular types of cells in the brain, we can actually observe something that really surprised me, which is that by injecting these complexes, we get very efficient editing in certain types of cells and really no editing in other types of cells. And we've done this in a number of different parts of the brain, and for time I'm just showing you a couple of examples here, but this is in the cortex. So over here on the left is a stain that just marks all of the cells, a sort of cell nuclei here. Then we're staining here with, in each case, a different antibody that recognizes proteins generated in a particular type of cell. Here is what we see when we look at this TD tomato reporter. So this is telling us where gene editing is occurring. And then this is a merge that is showing where these particular types of cells are located with respect to those where editing is happening. And what we found is that, so this is just a general stain for neuronal cells. Here we're staining for hippocampal striatal neurons, and you can see these individual cells here that are lighting up with the TD tomato, and when we merge these two images, you can see that there are actual editing events occurring in these particular types of neurons, compared to what we see down here, where we're staining for a protein expressed in astrocytes. And when we merge these two images, we see that there's really no overlap. So we don't see any evidence for editing in that type of cell in these tissue sections. So something that we're currently working on is to try to figure out how to do this even more efficiently. We've been able to engineer the Cas9 protein with cell penetrating activities, so it actually gets into cells a lot more efficiently than when we use the wild type form of the protein. And we're very hopeful that this approach will allow us to eventually do editing tissue selectively in other types of tissues as well. So let me now just turn very briefly to another topic that we've been interested in, which is namely, can we take our understanding of the Cas9 protein and use that information to allow us to create a different activity for this protein. And one of the things that we were intrigued by was the fact that in nature, this is a protein that is very selective for double-stranded DNA recognition, even though in principle you would think that it should be able to use its RNA guide to base pair with RNA molecules, for example. But we didn't see much evidence for that for the wild-type protein. And, but it was Mitch O'Connell and Ben Oakes in the lab who took on the project of trying to figure out if they could take advantage of what we understood about DNA unwinding by Cas9 to look at its interaction with other types of nucleic acids, namely with RNA. And so this is showing you an experiment in which we used DNA molecules that provide the PAM sequence to the protein in trans and use that basically as a trigger that binds to the protein, activates it for nucleic acid interaction, and then allows it to interact with single-stranded RNA substrates and generate a break. And I'll just show you very briefly that we've been able to now collaborate with the lab of Jean Yo at UC San Diego and his postdoctoral fellow David Nellis to use this for imaging particular RNAs inside of cells. And the attraction here is that one doesn't have to engineer the transcript. You can program the Cas9 enzyme to recognize an endogenous transcript and then visualize its behavior. And this is an experiment in which Jean and David were able to use this type of what we call rCas9 to track a particular transcript encoding beta-actin protein in cells. This is a control panel on the left that shows recognition of these clusters of sort of stress granule complexes in cells using an antisense DNA oligonucleotide. And then over here is using the rCas9 that's been programmed to recognize the beta-actin messenger RNA, which is localizing to these stress granules. And then this is using a stain for stress granules and then showing the merge over here. And control panels down here show you that we don't observe that kind of staining when we use a non-targeting guide for the rCas9 in these types of experiments. And I see the light flashing. Is that saying that I should? Is it time to stop? Is it time to turn it over to Dr. Zhang? Well, I won't. I'll just wrap up in a couple of minutes. Then I just want to say that this is a slide you've seen before, but basically in nature there are two types of CRISPR systems that naturally can bind and cleave RNA molecules called the type 3 and type 6 systems. And we've been investigating type 3 for a while, but type 6 caught our attention because here again, like with Cas9, there's a single protein that has RNA-guided RNA cleavage properties. This is actually a figure taken from one of a recent paper from Feng Zhang's lab in which this protein called C2C2, like Cas9, is programmed with guide RNAs, but in this case becomes an RNA targeting complex. And so we've been able to do experiments showing that this is a protein that actually has a second RNase activity. It's able to produce its own guide RNAs in a reaction that allows specific cleavage of precursor CRISPR molecules to generate these guide RNA sequences. And then this is a protein that becomes an RNA-activated RNA. So it's really an enzyme that has two RNase activities. I'm going to skip those slides and just jump to my conclusion here, which just summarizes that this protein here is an RNA-guided, RNA-activated, non-specific ribonuclease. It has a very interesting set of properties that we're actually now using as a very simple diagnostic that allows detection, very sensitive detection of RNA transcripts by activating this non-specific RNA cleavage activity. And it's work that will be published in a few days. So I just want to end by pointing out that I think it's very important to think about when there's a powerful technology that as we were discussing here, how do we use it? And in particular, in this case, what should we do now that genomes can be edited relatively easily? And I've been very involved over the last couple of years in thinking about this question and really inviting a global discussion around the ethics of certain applications of genome editing, in particular in the human germline, as well as for something called gene drives, which means driving a change through a population of cells or organisms very quickly. So I invite all of you to be involved in that discussion and to spread the word about genome editing. It's a wonderful technology. I think we want people to be excited about it, but we also want to approach it with appropriate caution. And I'll close by acknowledging a great group in my lab. This is taken at a recent baseball game, and we've had excellent collaborators that I mentioned along the way, and of course, extremely grateful for our financial supporters. Like Emmanuel, I just want to mention very briefly some company connections that I have. And I really just want to close by thanking Dr. Yin and the Tang Foundation for supporting fundamental science. It's really important. It's something that is really very important to me, and I think important for the future as well. So thank you very much.
Thank you, Jennifer. Our final speaker is Dr. Feng Zhang, or in Chinese, Zhang Feng. Dr. Zhang was born in Shijiazhuang in China and was immigrated to China, grew up in Iowa. And he received his bachelor's degree from Harvard University and PhD in chemistry and bioengineering from Stanford University. After that, he joined MIT, and he's now the W.M. Keck Career Development Professor in Biomedical Engineering in the Department of Brain and Cognitive Sciences and Biological Engineering. And like the other two speakers, Dr. Zhang has received numerous awards, primarily based on CRISPR-Cas9 technology, including 2016 Canada's Gardner International Awards, Human Genome Organization's Chen New Investigator Awards, Wilson Prize at Harvard University, and so on and so forth. Let's welcome Dr. Zhang.
Well, first of all, I want to echo what Emmanuel and Jennifer has said. It's really been a tremendous honor and a tremendous pleasure to be here. It's my first time here in Taiwan, but just based on the time I've spent here so far, I've already thoroughly enjoyed all the interactions that I had, and I really look forward to continuing these interactions in the next few days as we interact with students and colleagues. And I'm sure this won't be the last trip. And so some of those will probably blossom into continued collaborations where we can together push science forward. I also want to, more importantly, thank the Tang Foundation, Dr. Yin, and also the selection committee for giving us this tremendous honor. And so I thought before we dive into the science, we'll take a short break since we had two serious scientific talks. And I'll just tell you a little bit about where I started, how I got into this. So I was born in the 1980s in China, and growing up in China, mainland China, around that era, there was a huge emphasis on science education. My slides are not. Oh, there you go. There's a huge emphasis on science education, and so this is one of the pictures that I found on the internet. And you can see that there's really, you should love science, you should study science, and you should really use science. And this is really taught to us everywhere in school by talking to everybody in society as a way that we can push China forward to become a stronger, more modern country. So growing up in that environment, I became very interested in science very, very naturally. And also having engineering parents that kind of shaped my early interest and curiosity for science and technology. So the turning point really came in 1993 when my parents and I immigrated from Shijiazhuang, China, to Des Moines, Iowa. And so Des Moines is an interesting place. It's very different from China. So this is what China looks like. It's very crowded, full of people. And this is what welcomed me in Iowa. So I joke a little bit. So there's actually not just cows, but also quite a civilized place in China. In Des Moines. And I was very fortunate to have made that move because being able to study in the United States, studying in a different system where people or teachers really try to help you develop your own individual interests, that helped me develop my curiosity into biology, and also a lot of that shaped my curiosity and passion for what I do today. So when I got to Des Moines, I went to the Des Moines public school system. And so in addition to the normal classes in Des Moines, they also had Saturday enrichment programs, where one of the classes was on molecular biology. And so I didn't know anything about biology at the time. All I knew was that biology was about memorizing body parts. You would take paraformaldehyde fix little frogs or little rabbits and dissect and try to identify different organs and memorize what they are. That was not so logical or interesting to me. So in this class, they showed us a documentary. And this is a documentary that we watched. So from this movie, Jurassic Park, I got really fascinated about modern developments in biology, because in this movie, it not only taught us that biology, there's an underlying fundamental principle, but this fundamental principle, the way that DNA works, the way that DNA can translate into protein, and the way that the genetic code governs the basic rule of life, these principles we can use to be able to engineer biological systems and to do useful things. And that really appealed to me, because prior to that, I was very fascinated about programming and working with computers. And this sort of told me that maybe we can also program living systems to make our lives better. So my first break into science really came in high school. There was a local hospital called the Methodist Hospital, and they had a vocational program for students. So high school students would go there and volunteer in different departments: accounting, accounts payable, receiving, all these things that teach you useful life skills. But my teacher, the one who taught that enrichment class, told me that, well, besides working in these things where you learn useful life skills, there's also a gene therapy lab just in the corner of this hospital. You can really pick anywhere you want to work in the hospital. So since I knew you liked molecular biology, why don't you choose to volunteer in the gene therapy lab? And there, I really had my eyes opened. I had a fantastic mentor, Dr. John Levy, who was both a patient scientist, but also a very thoughtful molecular biologist. And he taught me all sorts of things that I had no idea about. And so many of those early experiments that he showed me how to do, those early fundamental biological principles, biochemical principles that he taught me, now I teach my own graduate students with. And I still use in my own experiments in the laboratory today. So after that, I went to college, and then my interest really started to develop into neuroscience, because I wanted to study a biological system that's mysterious, but also a lot of the diseases that concern the nervous system are ones that we don't understand, nor do we have the ability to treat. And so as I started to develop my interest into the brain, I enrolled in graduate school, and then that's where I learned how to do technology development. So I met my PhD advisor, Carl Deiserath, from a very serendipitous meeting where I was trying to find Steve Chu, who had moved to Berkeley just a few weeks before I arrived at Stanford. And Carl took over his office, but the sign on the door still said Steve Chu. So when I knocked on it, this white guy came out, and turned out that was Carl. So I was embarrassed, but I thought I should learn more about his research. And then through our conversation, he told me that this is an area, neuroscience, where we can develop really creative and powerful technologies to make the study of neurological diseases more tractable. So working with Carl and also Ed Boyden, we developed a technology called optogenetics, which uses microbial proteins called opsins. We can introduce them into specific brain cell types and be able to shine light, like using an optical fiber to be able to stimulate and modulate different brain circuits. And using that, we can start to understand how do signaling and how do circuits in the brain work. But as I was working on optogenetics, one of the core challenges had to do with how do we target opsins to specific cell types in the brain. One of the ways that we use in the laboratory was using transgenic mice that have Cre recombinases that are expressed in subtypes of cells, and we can cross that with a virus that's Cre dependent so that we can drive the expression of opsin in specific neuronal types. But to generate these transgenic animals where Cre has been inserted into specific locations in the genome is actually quite challenging. And I thought maybe genome editing, at the time, these older generations of technology like zinc finger nucleases can be adapted to make optogenetic an even more universal tool. As I started to explore the genome editing tools, I also realized that there's a lot more that we can do with it. Because as we sequence more and more human genomes, and especially the genomes of patients who have manifested forms of neurological diseases or other diseases, we are accumulating a large database of hypotheses of how genetic variation may contribute to different human disease types. And so here on the right is a plot from the US NIH, and it shows the number of publications that report genome-wide association studies. And you can see for the past 10 years, this has been growing exponentially. Each data point on this plot is a paper. And within each paper, oftentimes multiple genetic variations or multiple hypotheses have been advanced that suggest linkages between genetic variations and also disease pathology. But in order to make use of this data, we have to test the causality of these mutations. And so one way to do that is through using precision genome modification tools so that we can generate either cellular or animal models to be able to link particular variations to the disease state. So how do you go about making precise changes into the genome? Well, the genome is like a book. It's about 3 billion letters long. So if you were opening a Microsoft Word document, you would just go in and do a search and replace function and place the cursor in exactly where you want to make a change. But how do you do that in the context of the genome? Turns out there are alternative ways to do this, and these were worked out by scientists like Maria Jason and Jeff Haber, who realized that DNA breaks or cleavage sites within the genome are like cursors in Microsoft Word. Wherever you place a double-stranded break, cleavage is where you place the cursor, and that's where the editing activity can happen. And so in order to realize the ability to make these cleavage events and place the cursor in the genomic document, one way to do that is through the use of DNA binding domains. And so these DNA binding domains are oftentimes proteins that have naturally been evolved to be able to interact with specific sequences of DNA. But if we can modify the specificity of these DNA binding domains, we can use it as an anchor or as a search function and bring a nuclease which can be responsible for cleaving the DNA to a specific site that we want to edit. And so many groups have worked on developing these DNA binding domains to achieve this type of easily reprogrammable targeting specificity. So here are two of the earlier technologies. There are others as well. One of the more popular ones is called zinc finger proteins. And these are modular proteins that are found in most kingdoms of life. And they're modular in the form that each module can recognize 3 DNA bases. And you can link them together to form a zinc finger array. So if you want to target a longer stretch of DNA sequence, say 18 base pairs, you will simply put 6 of these domains together. And as scientists evolved the protein specificity, they were able to design the protein to recognize new DNA sequences. And because there are only 4 DNA bases, you would think that with 64 different modules, you will be able to target any DNA sequence you want. Turns out the theory is awesome, but in practice, it's actually quite challenging to be able to engineer a new zinc finger array. And that's because whenever you put 2 different modules together, they affect the specificity of their neighbor. And so it's not that you can put any 2 modules together and target exactly where you predict them to be. They actually target something different. So to make the system, to make DNA targeting simpler, there were other systems developed, and we and also others worked on modifying this bacterial pathogenic protein called tail to be able to recognize DNA. It's simpler than zinc finger proteins because it uses individual modules that can recognize single DNA bases. So you don't need so many, you just have to have 4, and then be able to put them together into a tail array to recognize the new DNA sequence. But building a tail protein, so tail proteins work better, more easily than zinc fingers, but they are still quite cumbersome to work with. And so this is when I thought, maybe we can try to develop a different system that will be easier, more affordable, and also faster to be able to target the genome. So when I first started my lab at MIT back in 2011, I read a paper by a Canadian scientist. His name is Sylvain Moyneux, and he had published a paper in Nature where he described that the CRISPR-Cas system, the one that contains Cas9, although back then he called it Cas5, is an RNA-guided DNA cleaving endonuclease. So wherever the RNA guide is in the cell in bacteria, shown here in the green, it binds to the DNA target site, and then it will cleave exactly 3 bases away from the 3' end of the DNA target site. When I read that paper, I thought this is really cool. If we can harness this system and then use the Cas9 nuclease and RNA to reprogram it to target a new genomic location, then we can be able to much more easily edit specific sites in the human genome. So around that time, in 2011, a lot of applications of the CRISPR system were actually focused on food production. So believe it or not, nowadays, almost 100% of yogurt that you use have been CRISPRized because the bacteria that's used to make these yogurt have been treated or being used CRISPR to become immunized against specific types of pathogens that might infect the starter yogurt culture. But we thought maybe rather than using them to produce yogurt, we can use them to do something different to be able to make changes within the genome of eukaryotic cells. So to get started, we set out on a 3-pronged roadmap to develop this technology. The first was to advance our understanding of the CRISPR system so that we can harness it and then figure out how we can use it to edit specific sites within the human genome. How do we use it to cleave the human genomic DNA? The second is once we develop this system, we wanted to see how well the technology can work in real-world context. And can we develop different ways to apply this to be able to study different forms of biological problems, whether it's in animal models or cellular models or in different types of screening applications. But just as important is the dissemination of the knowledge. And so the third prong of the roadmap is to share these reagents, to share the expertise, the protocols, and also as we improve the system, make the know-hows available to the broad scientific community. And so through a series of work, both by my group and also by other groups, now Cas9 is becoming a very powerful genome editing tool. And there are many ways to use it, and here are 2 of the main ways. The first is to use it as a way to edit specific sites within the genome. Using a RNA guide that has been reprogrammed to target a specific site, we can use it to cleave DNA, to place that genomic cursor, and then be able to edit specific sequences in the genome. But like Emmanuel and Jennifer mentioned, you can also inactivate these nuclease domains to turn it into a generic RNA-guided DNA binding protein. And this will allow you to recruit effector modules to a specific site. And this can be very modular. So whatever the effector is, be it a transcription activator or a transcription repressor or a fluorescent protein, you can use it to modify the transcription state from a given locus or be able to visualize specific sites within the genome. Besides these single loci applications, you can also use Cas9 in a much more high throughput way. And this is because to reprogram the specificity of Cas9 simply requires providing it with a different RNA guide. With the advance in DNA synthesis technology, especially with oligo-array-based oligo printing, we can now synthesize libraries of DNA oligos or these guides, 10,000, 100,000 of them at a time. And so using computation, using a computer algorithm that goes through the human genome, we can design RNA guides that can target every single protein coding gene within the genome. And then within a matter of few days, we can use oligoarray synthesis to print out all these guides and then clone the guides into viral libraries. And these viruses, lentiviruses, can integrate into the cells that we want to study. And so we can use this as a way of doing genetic selection. The way this works is we'll take a population of cells and we'll infect the cells with a lentivirus. We'll infect at the low multiplicity infection so that each cell usually only gets one or less virus. And so each cell will get a different guide RNA. But collectively in this population, we'll be able to modify, or we'll have cells that represent every single knockout event within these cells. So then we can apply a selection pressure. So for example, if you want to figure out what are the genetic mutations that would render a cancer cell resistant to the cancer therapeutic, we can apply onto this library of cells with different mutations the cancer drug like vemurafenib for melanoma. And then we can look to see what are the cells that are able to survive the drug selection. And so by sequencing the guide RNAs that are present in the surviving cells, we can infer what are the genes that, when knocked out, can give rise to drug resistance phenotype. You can apply the same type of selection or the same type of screen with other selections like viral resistance. You can look at different kinds of immune activation signals or different types of infectious disease resistance phenotypes as well. And there are many other applications too. So you can use this for both the loss of function screens where you use Cas9 to cleave the gene and inactivate it. Alternatively, you can use it to turn genes on. So by using inactivated Cas9 to recruit a transcription activator to a specific location, we can turn one gene on at a time and figure out which gene, when you turn it on, will give rise to a particular desired phenotype. So there are two ways to use this for doing genetic screens of protein coding genes, but most of the genome is actually non-coding. So how do you try to understand what are the functional elements in the non-coding genome? So it turns out that you can use similar kinds of screening methodology to figure out what are those regulatory elements within the genome. And so Neville, along with Jason in the lab, developed a way where they look at the non-coding elements that are flanking a particular gene region. And then by designing guide RNAs that target every single possible PAM within that non-coding region, they can generate a library. And then this library would allow them to perform saturation mutagenesis by introducing indel events at each one of these sites along the beginning or the end of the gene coding region. And so when you put this library of viruses onto the cell line that you want to screen with, you will generate a population that carry different non-coding mutations, and then you can apply the same type of selection pressure as you do for protein coding genes. So for example, we can take the same melanoma cell line, treat it with vemurafenib, the cancer melanoma drug, and then see what are the cells that will survive the vemurafenib treatment. And then by sequencing the guide RNAs and then mapping them to their target location in the non-coding genome, we can then figure out what are the functional regulatory elements. So this is what one of these experiments look like. COL3 is a gene that we identified from our previous screen, where if you knock it out or if you reduce the expression, it will render the melanoma cell resistant to the melanoma vemurafenib drug. And so we took the coding region based on the reference genomic sequence, and then we designed RNA guides that can tile along the 5' promoter region, or design guide RNAs that tile along the 3' region that is after the 3' UTR. And so each blue tick is a guide RNA, and you can see we can generate a quite high density mutation within these non-coding regions. So then we can perform a screen. And this is what the raw screening data would look like. So each dot here represents a single guide RNA, and then the red ones are the controls, and then the gray ones are, the red ones are the experimental, and then the gray ones are the controls. And you can see that there are certain guide RNAs that become enriched. So just looking at it this way doesn't really tell you very much. So the other way to look at it is to plot the enrichment frequency along the length of the 5' or the 3' non-coding region. And this is what the data looks like when you plot it. you can see that there are very specific locations where certain guide RNAs become, or cells receiving certain guide RNAs become really highly enriched after the drug selection. And so these suggest that there may be specific non-coding elements that regulate the expression of the COL3 gene. And so if you zoom in and look at each one of these regions, you will see that they correspond, some of them correspond to known transcription non-coding regulatory elements, and the other ones encode motifs that don't have known function, but likely indicate that they represent new, previously unidentified non-coding regulatory sequences. So if we zoom into this really large peak, you can see that there's a very large chip signal for the YY1 factor. And then also if you look at a particular sequence, you can very easily pick out the YY1 binding motif. And so if you go through these kinds of screens, you can quite readily identify non-coding regulatory elements for any gene that you're interested in learning more about. So these are some of the applications of the CRISPR-Cas system. You can modify a single gene, or you can do genetic screens, but there are also many other tantalizing possibilities. And one of the really important potential for this technology is developing it into a human therapeutic. We have now identified many different genetic mutations, more than 6,000 of them, that are linked to specific genetic diseases. And if we can go into the cells and replace those mutations through gene editing, then we can potentially have a broad spectrum treatment strategy for many inherited disorders. And so for the rest of the talk, I thought I'll share with you some of our work on further pushing Cas9 to become a therapeutic modality. And then the second topic is looking beyond a little bit more, looking into the bacterial diversity where many of these tools have came from, and then try to see where can we harness and develop the next generation of technologies. So let's look at the first therapeutic applications. So the S. pyogenes Cas9, which is most commonly used Cas9, is a large protein, is over 1350 amino acids long. And so that makes it quite difficult to package into viral vectors, which are commonly used for gene therapy delivery purposes. But if you look at the family of Cas9 proteins, there are different versions of Cas9s in different bacterial species. They come in different lengths. And so if you do a simple blast search in the sequenced bacterial genomes, you can identify many different bacterial species that carry Cas9. And if you plot them according to their length, you see that they distribute into two main clusters. There's one large cluster where the S. pyogenes Cas9 falls in. That's over here. And then there's a smaller cluster where another Cas9 that we developed very early on from S. thermophilus fall into. The Cas9 from S. pyogenes works quite well. S. thermophilus also works, but it's not as active as the S. pyogenes Cas9. So we thought maybe if we dig a little deeper into this family of small Cas9s, maybe we can find a new one that's both small but also quite active. So after a series of analyses, we identified one of them, and that's the one from Staphylococcus aureus, this particular one. And this is quite small. It's only about 3,100 base pairs long, and that makes it possible to package both SA-Cas9 as well as the guide RNA and all of the regulatory elements into a single adeno-associated virus vector. We focus on AAV because AAV is one of the most advanced gene therapy vectors, and it has already been approved for human use. And so we designed a AAV carrying SA-Cas9 and the guide RNA to see how well could it work in a mouse model to achieve editing in post-mitotic cells in a therapeutic context. And the first gene that we chose to target is called PCSK9, which is a very prominent drug target for reducing cholesterol and treating and reducing cardiovascular disease risk. So what we did is we packaged SA-Cas9 with a promoter that targets liver cells because that's where PCSK9 is produced. And then we also designed an RNA guide that can target the PCSK9 gene. So we built the virus and then we injected it through the tail vein, so it's a systemic administration of AAV. And then we try to see how well is the editing efficiency. So to analyze the level of mutagenesis, we took a punch from a piece of liver tissue, and we grind it up, and then we try to sequence and measure the amount of indel mutation at the PCSK9 locus. And so on the left side here is the editing data. So we injected 2 different viruses separately, and then they carry 2 different guides that target the PCSK9 gene. And so for animals that receive one or the other virus, we see similar levels or similar time course of editing event. So about 7 days after injection, you see editing approaching 50%. And then this is a permanent event. So once the cell has been edited, the mutation persists and stays in the cell. And so the percentage of cells that remain edited lasts for the rest of the experiment. And so if you measure at 14 days or 28 days, and now we'll have data that extend beyond the several months up to a year, we still see very good editing activity within the liver tissue. So there's good editing. We next thought to measure the amount of PCSK9 protein level in the serum of these mice. And so we took some time points that were before the injection and then also different time points after injection. And what we found is that the editing is quite robust, and the editing translated into a complete depletion of PCSK9 protein from these animals. And so here we injected 4 different dosages of the PCSK9 targeting SA-Cas9 virus. And then you can see that within 7 days of injection, you basically drop the PCSK9 protein level to undetectable levels. Whereas if the mouse has been injected with a control virus, you can see that they basically remain unchanged. And so this is very promising. It showed us that we're able to make indels in PCSK9 and also be able to deplete the protein level at really high efficiency in mice. Next, we measure the amount of cholesterol because a reduction in the expression level of PCSK9 in the serum should translate into a reduction in the cholesterol amount. And so you can see, indeed, we get a 50% reduction for the mice that were treated with Cas9, and then for the controls, they remain unchanged. And so this is very promising. It shows that the system can work in vivo, we can deliver it, and we can get to an editing efficiency level that's relevant for a therapeutic outcome. But this is just mice. So in order for us to move forward, we also have to test it in larger animal models, and then eventually we need to do very careful testing on human patients. So this is good, but there are many considerations that come into play when developing a therapeutic. And one of the major one is off-target activity. So we know we can achieve editing at the on-target site at PCSK9, but is it only making changes there, or is it making changes elsewhere in the genome as well? And so we and also many other groups have performed specificity analysis for Cas9, and we found that Cas9 can sometimes make off-target activity. And that's not ideal because we want to minimize other forms of genomic modifications so that we don't have to worry about potential confounds and also potential side effect consequences from these other edits. And so then one of the major challenges is to make Cas9 more specific. And so we try to make Cas9 closer to therapeutic grade. And so one of the approaches that we took is through a structure-guided engineering approach. And so Ian and Lingyi and Björn are 3 members of the lab that looked at Cas9 and thought about, based on what all of the research groups around the world have reported, the way that Cas9 binds to DNA. And they came up with a hypothesis of how they can engineer Cas9 to reduce off-target activity. So here is a schematic of how Cas9 binds to the target DNA. So the protein is here, and these are the 2 nuclease domains. The DNA is in red, and the RNA guide is in green. So in order for Cas9 to bind to a target site, the DNA, which is in duplex form, has to be unwound so that the RNA can base pair with one of the DNA strands. But when that happens, the other strand of DNA becomes destabilized. And so what that means is it's loosely hanging and it wants to re-anneal with the target DNA in order to be able to sort of leave and go to some other place. And so what we thought to do is that in order for this strand to be stabilized, there must be a region on the protein that's positively charged that can keep it bound at the target site. And so when we look at the crystal structure, this is what the structure looks like, colored in the same way as the schematic, but you can color it a different way by looking at a surface charge. And so here, dark blue means positive charge, and red means negative charge. And what you see is basically this groove where that displaced DNA strand is supposed to fall is highly positively charged. And that told us that, well, maybe we can try to neutralize some of these positive charges so that when the RNA is not perfectly bound to the DNA, this displaced DNA strand is not as stabilized so that it will compete with the RNA and then eject the Cas9 so that it doesn't make off-target activity. So we went through and we tested several dozens of mutations where we converted arginine or lysine, these positive charge residues, to a neutral residue like alanine. And eventually we found that certain combinations can significantly increase specificity. So here is data from one of the guys that targets VEGF-A. So based on genome-wide analysis, we found that there are 2 off-target sites with the wild-type Cas9. So you can see robust on-target editing, but also quite significant off-target editing. When we tested some of these mutants, we found that there are certain combinations that we call enhanced Cas9s that preserve the on-targeting activity and eliminates the off-target activity, at least using the methods that we were using to be able to detect these off-target modifications. And so using this methodology, we were able to then take one step to make Cas9 specific. But as we push the technology closer to therapeutic applications, we have to try to make it even more specific, because this is only evaluated using a certain level of sensitivity for off-target detection. And as we treat more and more cells in the body, we'll have to increase the sensitivity of off-target detection. And that also means that we'll have to make the system even more specific. So finally, I'll just shift gear and tell you about some of the ongoing work that we're performing to be able to further develop the genome editing toolbox. Because as we look into the bacterial diversity, there are many more systems that can be harnessed that can potentially have powerful biotechnological applications. We started by looking into the CRISPR diversity. So there are many different kinds of CRISPR systems. Both Emmanuel and Jennifer have showed you the distribution of different types. Largely, they can be classified into 2 main subtypes. There's class 1 and there's class 2. The main difference is that class 1 uses multiple proteins to do what class 2 can do using just a single protein. Cas9 belongs to class 2, and then at a time when we started this, there weren't sort of characterized, or there were not other class 2 systems that had been characterized. So we looked at one of them, and this is one called CPF1 that's found in a bacterial strain called Francisella novicida. And in Francisella, there are 2 CRISPR loci. There's one that encodes Cas9 and all the other components of the Cas9 system. But then there's a second locus that carries a similar kind of organization as Cas9, but the large protein is significantly different. And if you do bioinformatic analysis of the domain organization, you can see that it has a very different domain organization compared to Cas9. And so by putting the system into E. coli and trying to express it and through bacterial genetics and also biochemistry, we found that CPF1 is indeed a new type of CRISPR RNA-guided DNA endonuclease. So unlike Cas9, which is dual RNA-guided, it uses both a cRNA and also tracer RNA that Emmanuel told us about. CPF1 uses just a single RNA guide and is arranged in the mirror image as the Cas9 guide because the repeat element is now on the 5' end. It also cleaves DNA in a different fashion. CPF1 cleaves in a sticky end fashion, and Cas9 cleaves in a blunt end fashion. And this directly translates into differences in the indel editing result. These sticky ends often result in large deletions, whereas the single blunt indels result in small insertions or deletions. They also have different specificity profiles right out of the box. So here we designed 2 different guides, one that can be targeted by CPF1 because it has the 5' PAM sequence. And then it can also be targeted by Cas9 because it has the Cas9 PAM sequence on the 3' end. And using a method that we collaborated with Nicola Corsetto, we found that Cas9 has some off-targets, and CPF1 didn't exhibit the same off-target activity. So my time is running up, so I'm going to just skip the next couple of slides. But and I'll just say that as we start to perform additional computational analysis, we can now find new candidate genes that are potentially useful CRISPR effectors. And so here's Cas9 CPF1, and then more recently with Eugene Koonin and Konstantin Svirinov, we had reported the discovery of C2C1, C2C2, and also C2C3. And then most importantly, C2C2 contains new domains, like Jennifer mentioned, that can allow it to use RNA guide to target RNA. So finally, we're continuing to develop around the three-prong strategy, but also we're excited to apply it. But finally, just one point I want to mention, which is that as we look at the number of genome sequences from different bacterial diversity that are accumulated, this is really, we're only starting to look at the tip of the iceberg. Because as we sequence more and more genomes, we're going to identify many more bacterial enzymatic systems that can potentially have very useful properties. And we're really excited to continue to harness and develop the toolbox. So finally, I just want to acknowledge my group. This is really the most important slide. These are my students and postdocs who worked with me over the past 6 years to develop the CRISPR-Cas system for genome editing. We have also had a number of collaborators around the world who worked with us on developing various applications of the technology. And then also, of course, the funding agencies that supported our continuing work in this area. So thank you very much for your attention, and it's a great honor to be here.
Thank you, Dr. Zhang. Now we have heard three fascinating stories about CRISPR-Cas9. I think for those of you who have done the genomic modifications in the 1980s and 1990s, you know how difficult it was. At that time, we called it genetic engineering. And notice that this word engineering has never been appearing in the three talks in the above. Now we don't call it engineering anymore, we call it editing, meaning that it's so easy, you don't need an engineer to do it. So we can see the contribution of the three awardees and the power of this CRISPR-Cas9 it brings to us. More importantly, I wish all the young scientists will recognize that this curiosity-driven work can quickly lead to very practical applications and beyond. So let's thank the three awardees for the fascinating speech. Thank you very much, and thank you all for attending this afternoon's Laureate lectures. Thank you.
Thank you very much, Dr. Liao, for hosting this session for us. Please kindly remain on the stage. Now we would like to invite our three laureates, Dr. Charpentier, Dr. Downa, and Dr. Zhang, and also the CEO of Tang Prize Foundation, Dr. Zhenchuan Chen, to step onto the stage for a group photo shoot to remember this very special event. Thank you very much. Ladies and gentlemen, please give them another big round of applause. Thank you for their lectures. Thank you very much. Ladies and gentlemen, this is the end of the Biopharmaceutical Science lecture session. Thank you again for your participation. 各位来宾，第2届唐奖得奖人的演讲《生技医药厂》到此圆满的结束，非常感谢您今天的参与。离场的时候，请记得要将您的同步口译接收器归还到您之前所租借的服务区，并且要记得您随身携带的物品。祝您有个美好的午后，谢谢。